steminins 12 years ago. Right from the moment when we discovered it and developed it and started to use it and see the benefit that it was giving to some people, we also saw that some other individuals, it did not give all the benefit that we would expect. So we tried to start to understand why is this. And it led to the understanding or the hypothesis that something, it's okay, I have 3 glasses here, but thank you. I will start with yours. Thank you very much. So the idea was, what is it? It's the blood that delivers cells to all the parts of the body. The blood is your transport system. If you have problem in blood circulation, that means you have problem in distributing stem cells in the body. So the question is, what can prevent or hinder or reduce blood circulation, and what can we do about it? That's really what was behind the development of StemFlo. So to explain what we have developed, let's just talk a little bit about cardiovascular physiology. So your blood leaves the heart, goes into arteries, arterioles, capillaries, and then after that, small veins, goes to the lung, gets oxygenated the heart, and it goes like this into the whole body. The only place where all of this matter is at the level of the capillaries, because it's only at the level of the fine capillaries, extremely fine, much finer than a hair, extremely fine, so fine that they are everywhere in your body, and you don't have more than, like a cell is never more than 3 cells away from a capillary. That is how many capillaries you have in your body. If you put them all side by side, or one after the other, you would go to the moon and come back. You have, literally, you have a lot of capillaries in the body. They're so small, it's at that level that oxygen, nutrients, minerals, vitamins, everything leaves the blood circulation to go and feed the cells. The rest is only plumbing. It's piping. It's how you bring the blood to those capillaries. And yet, if you go see the doctor, they never look as to whether the blood goes where it needs to go. What they do is that they take the pressure in the big artery, because if the pressure is too high, it can lead to heart problems. And they take a blood sample and they tell you what your blood is made of. If it doesn't get where it needs to go, it's never looked at, as long as it contains the right thing. I'm not saying that it's not relevant to look at the blood composition. It's very important. Very important to look at the blood pressure as well. But also extremely important to make sure that transport system can really bring everything everywhere in the body. Nutrients, minerals, everything, but also cells. The cells must be able to circulate in all these capillaries. And that is really what is crucial here. So let's look at all of this with a different picture here. So the heart, arteries, arterioles, capillaries, vein, and then back to the heart. The reason why I want to show this, the reason why I'm talking about all of this is that when we talk about cells, cells cannot leave. All of your blood vessels are fairly thick. They have muscles around them, arteries, arterioles. They don't allow cells to leave the blood circulation across these big physiological, anatomical walls. The only place where a cell can leave the blood circulation and go into a tissue is in a place called the post-capillary venule. And here's what happened. What is the difference between a capillary and a vein? The main difference, sometimes people think it's size, it's not, false, but the main difference is that capillaries have muscles around them, veins don't have muscles around them. So it's pressure. And it has a very important role in the body, because the stem cell has specific molecules on its surface sensing shear force. Okay? So I'm a stem cell, and I'm circulating right now in high pressure capillaries. Suddenly I fall into low pressure venule. Right at that junction, what happens when you go from high pressure to low pressure? It's like a river that rushes into a calm lake. Right at the entrance, you get vortices. You throw a piece of wood and it goes counter current. So I'm the stem cell circulating and I get into this vortices. So these molecules here are bent right and left. These trigger the expression of receptor that makes the stem cell listen to the tissue. Is SDF1 coming around? Yes, it is. I grab the capillary. It's exactly what happens. So that's the area where stem cells get responsive to tissue and they can migrate into tissue. The reason why I'm saying all of this is that if stem cells cannot go there, it doesn't matter if you release them. Think of all of this as the train system in Malaysia. You want to go to Ipoh, you get on the train, you arrive in Ipoh, the door don't open. What was the purpose of getting on the train? You wasted your time. So you release stem cells, if they cannot get into that area, it's pointless. So what is it that could prevent stem cells to go in that area? What is it that can reduce capillary flow? It's a process that was discovered in the 1990s called oxidative coagulopathy, coagulation. Coagulation that takes place when it should not. I'm not talking about blood clot. A blood clot is a real cascade of events that lead to the full formation of a blood clot. Here I'm talking about simply the effect of free radicals on a specific enzymes that will transform fibrinogen into fibrin molecule, which are sticky, they will stick together, and they will form a fibrin mesh. It's like a hairball in your drain after you finish your shower. It's not going to stop the flow of water, it's going to reduce it. But if there's anything floating in the water, it gets filtered by it. So you get a mesh of fibrin that can be sometimes 2, 3, 4, 5 times the size of the entrance of these capillaries, and it gets lodged right there. It sits like a hairball in front of these capillaries, and it filters the entrance of cells. You still have flow of nutrients, oxygen, these kind of things, but you've reduced the flow of cells. So if I have this right there, that means all the tissue fed by these capillaries are deprived of the opportunity to repair. So obviously, if we really want to give stem cells give them their full potential, we need to give them the ability of circulating everywhere in the body. It's that simple. So what did we do? We developed a product that contains enzymes that specifically digest fibrin in your blood. In order to be active, you need to take them away from meals, 30 minutes before a meal or 2 hours after a meal, so that they fall in the stomach, go in the intestine, the capsules open in the intestine, so enzymes can cross and get into the blood as active enzymes. And we have tested it in the lab. If somebody takes StemFlo, an hour later we take a blood sample, and the ability of the plasma to digest fibrin is very significantly enhanced. Since fibrin formation takes place in the presence of free radicals, we have a lot of antioxidants, natural antioxidants. Since it's a product about blood circulation, we've added a number of ingredients that are very well known to support the integrity of small blood vessels, small capillaries. And if you enhance capillary flow, you will increase. Remember, capillaries, you will have enough to go to the moon and come back. This is a lot of vessels. So when you start to open them, you will pool a lot of blood into these capillaries, meaning you increase the pressure in your tissues. You reduce the pressure in your arteries. And that also was documented in that clinical trial. You give StemFlo to people, within 1 hour, you take blood pressure before and after, and you have an average of about 5 millimeters of mercury drop. It's not huge, but it's just a clear indication here that the product is actually working. We actually also measured the oxygen being liberated by the skin, oozing out of the skin, as an indirect measure of capillary flow. And you have the person laying down, and after time, without moving, oxygen starts to decrease. Being freed from the skin, it decreases. You give them StemFlo, 20 minutes later, the oxygen starts to increase. So the product works very well. But when you increase pressure in the tissue, that means you also increase the amount of fluid that will leave the blood vessels to go into the tissue. So you increase lymphatic circulation in your tissue, so you increase cleansing of your tissue, which is very, very good, but it leads then to sometimes symptoms of detoxification. So we included things like curcumin to buffer. It's known, coming from turmeric, it's known to really buffer various aspects of detoxification. So this is, again, StemFlo, and this one, it took a long time to finally get the approval in Malaysia. It was finally obtained, and then the product is, as we speak today, in manufacture, right? So it should be very, very soon. Do I say the date that you gave me? Nick said no. So it will be before November, right? I've learned to be safe. Though it's going to be very soon. Okay? How soon, I can't tell, because Nick needs to still bring me to the airport tonight, so I can't tell. ST5. This one is in the process of registration. It may come a little bit later when compared to SC2 and StemFlo, but I'll talk about it nonetheless because it's part of all the scientific work that we have done. Remember what I told you? We started to look at a lot of different plants known to be associated with a broad variety of health benefits. Goji berry, for example, colostrum, medicinal.